Okay, gentlemen, good morning. Today is Monday, April the 27th, and today we are going to cover the last section of chapter 16, which is basically about the molecular structure of a chromosome and how proteins and DNA kind of wrap and around each other and hold everything together. So, you'll also be very pleased to know, I think, that this will only be one video. There's only 7 slides in this section, and so hopefully it won't take all that long. All right, that'll make us happy. Now, um, there are major differences between, and you might guess this, between prokaryotic chromosomes and eukaryotic chromosomes. Remember that in bacteria, the chromosome is a single chromosome and it's circular. All right, so just one big loop. It's still double-stranded, but it doesn't require a whole lot of packaging to keep it in its looped structure. So there is very little protein associated with the bacterial chromosome. On the other hand, in eukaryotes, which include the protists, the fungi, the plants, and animals, the DNA molecules are linear. And therefore, in order to keep them in their shape and together, there's a lot of protein around which the strands of DNA are actually wrapped. So, let's talk about the eukaryotic scenario here. The DNA is wrapped in a eukaryotic chromosome around special proteins, and they form a material together called chromatin. The proteins that the DNA is wrapped around are called histones, and each histone is comprised of 8 little proteins, 2 of each of 4 different types. And so what essentially happens, and you'll see this in the picture on the next slide, is that the long, thin strand of DNA wraps around those histones, and that's the primary structure of a chromosome. If you take a thick chromosome that you see during mitosis and meiosis, and you kind of unwind it like threads coming out of a sweater or something like that, you'll see that it looks almost like a beaded necklace. It's a long string with a bead here and a bead here and a bead here and a bead here. And so each bead is what we refer to as a nucleosome. And the nucleosome itself consisted of those 8 histones that we talked about a minute ago, with the DNA wrapped around them. And so that is that. There are ends of the proteins that stick out, so it makes it look like almost like a sea urchin with little spikes sticking out of it all. And so the centers of the proteins are tight in the middle, and then you've got these little protrusions, pointing out in different directions, and it allows you to wrap the DNA around them. And so this is what I'm talking about. Hopefully this gives you a better idea of what I was babbling about in the previous slide. So here's the double-stranded DNA, and this represents a histone. All right, you take 8 histones and you wrap the DNA strand around it twice, and you can see that very clearly in the area I've just circled. And this is what we refer to as a nucleosome. Now, the histones themselves pack themselves together, and you can see it gets much thicker. And so the DNA and the histones together are now almost 3 times the diameter of a single nucleosome, all right, or a single histone package, I would say. Now, that fiber is then looped around what we call a scaffold. And so there's a central scaffold that runs right down this, whoopsie daisy, right down this fiber. And you can see that this stuff is now found in loops here and here and here. All right. And then that is, for crying out loud. And then that is packaged into a chromatid, which which you can see here. All right. So that is the deal. That's what the structure of the chromosome actually looks like. As always, as I told you the other day, I have uploaded this PowerPoint onto, I think it was from last Thursday, maybe. I think it was, maybe it might have been Monday. No, I don't know. Actually, let me look. Should have been April 22nd, which is a Wednesday. All right, so go back and look at this animation in the PowerPoint so that you can see this a little bit more clearly. Now, the chromatin itself is not just randomly spread around inside the nucleus, all right? During interphase, the chromatin has kind of thinned out. You don't see the chromosomes that like you see here in the karyotypes. All right, the chromosomes themselves kind of expand, and so you can't see individual chromosomes. However, in a very clever experiment done with phosphofluorescence, a bunch of scientists actually color coded the various chromosomes, and so you can see, like, this area represents one chromosome, and this area represents one chromosome. And so at interphase, the chromosomes are not just all tangled up next to each other. They do actually occupy a very specific space within the nucleus. Now, during interphase, most of the chromatin is loosely packed, but it does condense prior to mitosis and to meiosis. The loosely packed chromatin is called euchromatin, which literally means true chromatin. But the very ends, where you find the telomeres, is actually called something different. It's called heterochromatin because it's much more tightly packed, which makes it very difficult for genes in that area to be expressed. Now, we're going to get into gene expression in the next chapter, but that's neither here nor there right now. Histones can result or undergo modifications that can lead to changes in the condensation of a chromosome. And these can also have effects on gene expression. Okay, so that, my friends, is the end of that, I believe it is. So here's the deal. Today is Monday, April the 27th. For tomorrow, April the 28th, I will post the multiple choice questions for chapter 16. Please go through those. And then for Wednesday, I'd like you to do the mastering for chapter 16. That will be due on Thursday at midnight. Okay, so you'll have a couple of days to work on that. All right, so that is the end of our discussion of chapter 16. Gentlemen, happy Monday.